Hello everyone, I am Dr. Nilanjana Bhattacharya Nath from Swami Vivekananda Institute of Modern Science. Today my topic is bacterial conjugation. So in this video, I will be discussing about the basic stages of bacterial conjugation. Let's start with a brief introduction. Bacterial conjugation was first described in the year of 1940 by Joshua Lederberg and Edward Totham. Conjugation is the process by which one bacterium transfers genetic material to another through direct contact. During this process, one bacterium serves as the donor of the genetic material and the other serves as the recipient. The donor bacterium carries a DNA sequence called the fertility factor or F factor. The donor cell, also denoted as F plus, and the recipient cell is known as F minus, which receives the plasmid DNA from the donor. The F factor allows the donor to produce a thin tube-like structure called the pilus, also known as sex pilus. And this sex pilus joins the donor with the recipient during this gene transfer process. Next, different stages of bacterial conjugation. Basically, there are 4 stages. Number 1, mating pair formation. Number 2, conjugal DNA synthesis. Number 3, DNA transfer. And the last, that is maturation. In this slide, the diagram showing the different stages of bacterial conjugation process as I have told you. Now the different stages. Number 1, mating pair formation. Sex pilus is a long, thin fiber that extends from the donor bacterial cell surface. About 20 genes of ORIT. ORIT stands for origin of transfer. That means from that particular site, the plasmid DNA transfer will start, and the F factor are required to produce a functional pilus that is also known as sex pilus. F plasmid carries Tra A and Tra B locus, which are about 33 kilobase pair long and consist of about 40 genes. The Tra locus includes the pillin protein, basically the pillin gene. After the transcription and translation, it synthesizes the pillin protein and other regulatory genes which together form pili on the cell surface. Next, conjugal DNA synthesis. Conjugation is initiated by a signal. The relaxase enzyme creates a nick in one of the strands of the conjugative plasmid at the ORIT site. Basically, that DNA synthesis is also known as the rolling circle replication process. By this process, the plasmid DNA will synthesize its new DNA strand, and that particular DNA strand is transferred from the donor bacterium to the recipient. And the rolling circle model of DNA replication basically consists 5 different steps. Number 1, circular double-stranded DNA will be nicked. Next, 3' end is elongated and also acts as the leading strand. 5' end displaced. It acts as the lagging strand, made up of a double-stranded fragmented DNA molecule that is also known as Okazaki fragments. Next, replication of both unnicked and displaced single-stranded DNA take place. Lastly, displaced DNA circulates and synthesizes its own complementary strand. In this overall conjugal DNA synthesis, relaxase may work alone or in a complex of a group of proteins known as relaxosome. The relaxase enzyme is also called Tra-I, and the relaxosome consists of Tra-I, Tra-Y, Tra-M and the integrated host factor, that is IHF. Next, DNA transfer and maturation. The nicked strand or T strand is unowned from the unbroken strand and transferred to the recipient cell in 5' to 3' direction. Only the DNA strand that contains the nick is transferred to the female cell. DNA replication of the transferring strand in the female cell and the single-stranded DNA that left behind in the male cell ensures that both cells have double-stranded DNA. Lagging strand synthesis occurs only in the female or recipient cell, while leading strand synthesis takes place only in the male or donor cell. Once the affected DNA has been moved into recipient cell and undergone replication to double-stranded DNA, it is established as a plasmid, the same as in the donor cell. Next, horizontal gene transfer. Horizontal gene transfer is a process in which prokaryotic cell can acquire genes from other microbes of the same generation, which in some cases can be a different species or even a different genus than the donor. Therefore, they use conjugation to exchange DNA between two different bacteria. It actually involves two bacteria. One bacterium extends a pilus to the other bacterium, and as I have discussed, that particular bacterium is known as the donor bacterium or the male bacterium, and the other is denoted as recipient or female bacterium. So the important question is bacterial conjugation the same as the sex in eukaryotes? Conjugation is equivalent to sex in that it combines genetic material from two different individuals into a single genome. Like sexual reproduction, conjugation produces new genetic combinations. Sex involves the vertical transfer of genes. Genetic information is passed down vertically from parents to their offspring. But in conjugation, it is not involved in reproduction as no new individuals are produced. So conjugation is an example of lateral or horizontal transfer of genes.